In this video, we will talk about a little bit more about what genetic diversity actually is and how Hardy Weinberg equilibrium is one way that we can use to assess genetic diversity in a population. So what is genetic diversity? In terms of the actual definition, genetic diversity of a population refers to the numbers of different alleles that are contained within all individuals. And if we look at this example here with the different corn, the genetic diversity is what's responsible for these different colors of kernels. So we have some that are red, we have some that are yellow, some that are black, etc. And the reason why I emphasize the genetic diversity is the alleles contained within all individuals is that if we look at this picture, for instance, if by chance we only sampled the yellow corn, we're actually not capturing all the genetic diversity of this population because they don't contain the dark red color that's here, right? And so when we're thinking about genetic diversity of a population, it's referring to the number of alleles present in all individuals. So the red alleles in the red corn, the yellow alleles in the yellow corn, etc. So it encompasses all of that overall within all individuals. And as we talked about in the module overview with the koalas, genetic diversity is so important because it's needed for populations to be able to evolve and adapt to changing environments, which of course is very prevalent in today's world. The example on the left is a koala that I talked about previously. The example on the right is another example of a species or population that is really threatened by low genetic diversity, and that's actually the bananas that we eat. So we eat a variety of bananas called Cavendish bananas. All of those bananas are actually genetic clones. They're genetically identical, and a lot of the plantations or farms that grow these bananas are suffering from a fungal disease. So I believe this is a picture from Central or South America, and a lot of these bananas are being attacked by this fungus because they have really low genetic diversity. They're not going to survive. So if we think about how this relates to our everyday life, if you eat bananas like I do, I have some in my kitchen right now, that variety of bananas actually might not be able to survive because of its low genetic diversity. Okay, so when we think about populations, and one of the ways we can quantify the genetic diversity that it has is think about its allele and genotype frequencies. And one of the ways we can calculate genotype frequencies, if we know allele frequencies, is we can calculate that based on these equations, and that will tell us whether a population is in Hardy Weinberg equilibrium. So let's take a step back here and just make sure we all remember these definitions here for genotype and allele. So let's use different colors. If we're talking about alleles, those are the different gene variants, right? So if we're talking about flower color, we might have a capital A allele, a lowercase a allele, right? Those are the two different alleles. And then the genotype frequencies, let's use a different color. Refer to those different combinations of alleles. So we have three different genotypes basically, right? And so there are equations we can use to figure out, based on the allele frequencies, what we would expect the genotype frequencies to be if a population is in Hardy Weinberg equilibrium. There's a lot of section in your book that works through how to actually calculate this, in box 5.1, for example. I'm not going to ask us for our purposes to know how to calculate this because there are ways to do it in computer programs in R, for instance. But what we'll focus on here is knowing how to interpret the results of that. So that's the genotype, difference between the allele and the genotype frequencies. If a population is in Hardy Weinberg equilibrium, or I'll often abbreviate that as HWE, then the expected genotype frequencies that we calculate from the allele frequencies will match the observed genotype frequencies that we actually see in the population. So we basically do it one way, just computer calculations, and then the other way is looking at what we actually see in the population. How many of each of these genotypes do we actually see? And the expected and observed genotype frequencies, however, will not match if the assumptions of Hardy Weinberg equilibrium are not met. So the assumptions are listed here on the right in cartoon form. This is not mine, but I appreciated it and thought it showed things very clearly. So when we're using those equations to calculate genotype frequencies from allele frequencies, we're assuming that individuals are mated randomly. We're assuming the population is really large. We're assuming that no selection is happening, that no mutation is happening, and there's no migration happening. There are no individuals moving between populations. If those conditions, or some or all of them, are not met, that means that the observed genotype frequencies that we see and the expected genotype frequencies that we calculate are not going to match. And how do we tell if they match or not? We do a test called a chi-squared test, which again, we don't need to know how to actually do the hand calculations for this because you can do it on a computer, but it is important to know how to interpret it. So when you do a chi-squared test, you get a p-value. If your p-value is less than 0.05, that means there is a significant difference between your observed and expected. So you would say that population is not in Hardy Weinberg equilibrium. If the p-value is greater than 0.05, that means those two values are not significantly different, so we would say that it is in Hardy Weinberg equilibrium. So again, what I'm focused on for our purposes in this class is for you to be able to understand how to interpret these chi-squared values and what that means in terms of Hardy Weinberg equilibrium. And really, if we do find that populations are not in Hardy Weinberg equilibrium, that's actually where things can get kind of interesting. And what we want to do from a molecular ecologist standpoint is figure out why that is. So again, if we think back to some of those assumptions, we said there's no selection happening and that the population is infinitely large. Well, really, if some of those conditions are not met, so if we do have selection happening or if we have inbreeding happening, that's actually kind of interesting. So I remember when I was doing my dissertation work, I had some data and I went through it, figured out which loci, which genes were not in Hardy Weinberg equilibrium, and I threw them out. And I was meeting with my advisor and he was like, well, why did you do that? And I was like, I thought that's what you were supposed to do. But really, if they're not in Hardy Weinberg equilibrium, especially for me when I'm interested in loci that are involved in adaptation, those are some of the really interesting ones you actually want to pursue further. And so when we're thinking about how we might actually do that, so how do we identify if loci are under selection or if inbreeding is happening? A lot of this we'll cover in more depth later in the semester, but I have two images here to illustrate that. So if we want to identify selection here on the left, this is a very busy plot, but in brief, each one of these blue dots is a different SNP that was identified through RAD-seq high-throughput sequencing. And all of the SNPs that are in this red region here are actually under selection between the Northern and Southern California populations I've been talking about. This is data with the intertidal snail that I work with. So that's one potential reason why the populations might not be in Hardy Weinberg equilibrium is if selection is happening on these interesting SNPs that we might want to figure out what genes those SNPs are in and what potential functions they might have. Another reason why populations might not be in Hardy Weinberg equilibrium is if something like inbreeding is happening. And I know this equation doesn't mean much to you, but it's just to illustrate there are ways that we can actually calculate what are called inbreeding coefficients to then figure out if our population in question, for instance, is really small and is suffering from inbreeding, like they found in elephant seals or Florida pumas, for instance.